DNA fingerprinting. DNA fingerprinting is a technique used to identify individuals based on their unique DNA patterns. It relies on detecting variations in specific regions of the DNA, particularly variable number tandem repeats, VNTRs, or short tandem repeats, STRs, that differ from person to person. Although 99.9% of human DNA is identical among all people, the remaining 0.1% differences, especially in non-coding regions, form a unique pattern, like a genetic barcode. Things required for DNA fingerprinting. Category: Items/materials required. Sample source: blood, hair root, saliva, semen, skin, bone, or any biological tissue containing DNA. Enzymes: restriction endonucleases, to cut DNA at specific sequences. Probes: radioactive or fluorescent DNA probes complementary to STR/VNTR regions. Gel material: agarose or polyacrylamide gel, for electrophoresis. Buffers and reagents: tris-EDTA buffer, loading dyes, ethodium bromide or fluorescent stains. Equipment: PCR machine, for amplification, electrophoresis unit, UV transilluminator, autoradiography setup, or imaging system. Software/database, modern use. STR analysis software, CODIS, combined DNA index system, or other forensic databases. Steps/process of DNA fingerprinting. Step 1. DNA extraction. DNA is isolated from cells present in biological samples, blood, hair, saliva, etc., using chemical or enzymatic methods. Step 2. DNA quantification and purification. The extracted DNA is purified and quantified to ensure good quality for analysis. Step 3. DNA fragmentation. Restriction enzymes are used to cut DNA at specific recognition sites, generating fragments of different lengths. Step 4. DNA amplification, using PCR. Specific DNA regions, STR/VNTRs, are amplified using polymerase chain reaction, PCR, to get sufficient DNA quantity. Step 5. Gel electrophoresis. The amplified DNA fragments are separated on a gel according to size. Smaller fragments move faster, creating a banding pattern. Step 6. DNA transfer, Southern blotting, traditional method. The DNA fragments from the gel are transferred to a nylon or nitrocellulose membrane. Step 7. Hybridization with DNA probes. Radioactive or fluorescent probes complementary to specific STR/VNTR sequences are added, which bind to the target DNA fragments. Step 8. Detection and visualization. Autoradiography or fluorescence imaging is used to visualize the hybridized DNA bands, creating the DNA fingerprint pattern. Step 9. Comparison. The DNA pattern, banding, from the unknown sample is compared with reference samples. Identical patterns indicate a match. Applications of DNA fingerprinting. Field. Applications. Forensic science. Identification of criminals from blood, hair, semen, or skin at crime scenes. Paternity and maternity testing. Determining biological relationships between parents and children. Identification of missing persons/disaster victims. DNA helps identify remains in accidents or natural disasters. Medical diagnosis. Detecting genetic disorders and carrier status. Anthropological studies. Tracing human ancestry, evolution, and migration patterns. Wildlife and conservation biology. Identifying animal species, tracking illegal trade of endangered species. Agriculture and breeding. Verifying plant or animal pedigree, ensuring genetic purity in breeds. Modern techniques in DNA fingerprinting. Traditional method. Modern method. RFLP, restriction fragment length polymorphism. STR, short tandem repeat, analysis. Requires large DNA quantity. Works with small or degraded samples. Slow and time consuming. Fast, automated, high accuracy. Uses radioactive probes. Uses fluorescent tags and computer analysis.